Hello everyone. Today we will discuss about phytohormones or plant growth regulators or plant hormones. We will discuss its general characteristics, and today we will focus on two plant hormones, that is Auxin and Gibberellins or Gibberellins. So let's start. So first of all, what are plant growth regulators? So these are the chemical substances which are produced in very small amount, and they have their unique function in the plants. So these are active in very low concentration, and these are commonly known as chemical messengers also. और ये produce होते हैं certain parts of the plants, and they are usually transported to the other parts where they are required, जहाँ पे ये अपना function perform करेंगे. And they have elicit or specific biochemical, physiological or morphological functions or responses in a particular portion of the plant. So this picture shows the various functions of plant hormones like auxin, gibberellic acid, or this cytokinins. They helps in stem elongation or cell elongation or growth of the plant. Then there is another hormone, that is abscisic acid. That is a stress hormone which helps in closure of stomatals and prevent water loss. Then there are other hormones like ethylene and auxin. Their ratio decides the formation of this abscision zone. Then we have other type of hormones like ethylene, which is the gaseous hormone and helps in ripening of fruits, and cytokinins that helps in cell division and growth of plant parts like leaves, fruits and stem. Then auxin also helps in formation of roots, so that phenomenon is commonly known as gravitropism, which means movement towards earth. So these are some functions of plant hormones in plants that we will discuss one by one in detail. And these plant hormones which are produced in certain parts, they are also functional in that part. So they are also active in the tissue where they are produced. जहाँ पे produce होते हैं वहाँ पे भी ये functional होते हैं. So these substances which are active in very small amounts, these are commonly known as plant hormones, phytohormones or growth regulators. Next is characteristics of plant growth regulators. So first of all, these plant growth regulators are the small, simple molecules and having the diverse chemical composition. So chemical composition इनकी vary करती है, and they could be indole compounds. For example, we have one important hormone, that is auxin, oxaloacetic. This is indole 3 acetic acid. Then we have derivative hormone, a derivative of adenine hormones like this Kinetin, that is furfuryl-amino purine. Then we have derivatives of carotenoids, basically the sesquiterpene, that is of 15 carbon, that is abscisic acid, and the diterpenes like gibberellic acid. It contains 20 carbons. Then we have some gaseous hormones like ethylene, which helps in fruit ripening. So there are 5 well-known hormones like auxin, gibberellin, cytokinin, ethylene and ABA. Out of these 5, these first 3 are considered as growth promoting hormones, and rest of the 2 are considered as growth inhibiting hormones. There are some chemical substances which are recently reported like steroids, salicylic acid, jasmonic acids, which function just similar to that of other hormones. So they are also considered as hormones. So the PGRs are active in very low concentration, usually the concentration of 10 raised to the power minus 6 molar. Next, classification of plant growth regulators. Broadly हम क्या 2 category में इनको divide करते हैं. First in natural and another synthetic, that is man-made. Natural hormones are of 2 types. First, plant growth promoters, means which enhances the growth, that is auxin, gibberellin and cytokinin. Then growth inhibitors, which will prevent the growth, these are ethylene and abscisic acid. Then in synthetic plant growth regulators, so these are the synthetic auxins like NA, that is ethylene acetic acid, 2,4-dichlorophenoxyacetic acid, and 2,4,5-trichlorophenoxyacetic acid. So this is the outline of classification of hormones. Next we will start with auxin, commonly known as the master hormone. So the term was coined by F.W. Wendt, and auxin in Greek means auxin. This is derived from Greek word auxin, which means to grow. So it helps in growth of the plants, and it was first isolated from the urine of a patient, and it was the first hormone to be discovered. ये पहला हार्मोन है जो डिस्कवर हुआ था. And the term auxin applies to a heterocyclic compound, that is indole 3 acetic acid. So this is the picture of this indole 3 acetic acid. This is the indole ring containing this acetic acid group. It is mostly produced or synthesized in the growing parts of the plant, mostly in the apices of stem and roots, और वहाँ से ये migrate होता है उन parts में जहाँ पे इसकी requirement होती है, and migrate to the regions of their action, जहाँ पे ये अपना function show करता है. So mainly it helps in promoting the cell elongation in stem and coleoptile. Coleoptile is the embryonic shoot of some grasses. Next is discovery. So in this field, firstly, Charles Darwin and his son Francis Darwin in 1880 noticed that जो यूनिलेटरल लाइट है उसकी वजह से जो grasses है मोस्टली जो उन्होंने स्टडी किया था, दैट वाज कैनरी ग्रास, दैट ग्रास शोज कर्वेचर इन द डायरेक्शन ऑफ लाइट. मूवमेंट शो करता है इन द डायरेक्शन ऑफ लाइट. And the botanical name of this grass is Phalaris canarensis, and this grass coleoptile show curvature in the direction of light. So by this picture we'll understand this. So this is the coleoptile of this grass, that is Phalaris canarensis. जब यहाँ पे unidirectional light दी जाती है, so ये जो coleoptile है it shows curvature. This seedling show curvature in the direction of light. When they excised or decapitated this apex, जब ये coleoptile यहाँ से remove किया and और ये जो light इसमें पड़ी, so this seedling was unable to receive the stimulus or perceive the stimulus, और इसने इस light के direction में curvature show नहीं किया, growth show नहीं की. Again when this tip is covered with some foil, तब भी ये जो seedling है it is unable to sense the stimulus of light है, और ये कोई भी curvature show नहीं करता है. So they observed from this experiment that this tip is the source of chemical substance जो कि टिप से सब एपिकल रीजन में आता है, और this is the responsible for this unilateral growth in the direction of light. They decapitated coleoptile, and coleoptile with its tip covered with foil. So these in two conditions, जब coleoptile को decapitate means remove किया जाता है, और दूसरा coleoptile जो tip है that is covered with some foil, so this seedling is not able to perceive the light stimulus. तो ये seedling light stimulus को perceive नहीं कर पाता है, और ये curvature को show नहीं करता है in the direction of light. Next experiment was carried out by Auxin Janssen in 1913. So इन्होंने ये ऑब्जर्व किया जो फोटो पीरियोडिज्म पिक करता है कोलियोप्टाइल, जो लाइट स्टिमुलस को रिसीव करता है कोलियोप्टाइल, सो इट इज़ ट्रांसमिटेड टू द सब एपिकल रीजन. ये एपिकल रीजन से सब एपिकल रीजन में जाता है थ्रू द ब्लॉक ऑफ जिलेटिन बट नॉट थ्रू द मिका प्लेट. सो जिलेटिन के ब्लॉक से ये सब एपिकल रीजन में जा सकता है, बट मिका प्लेट के ब्लॉक से नहीं जाएगा. सो व्हाट दे हैव डन, इन्होंने जिस डायरेक्शन में लाइट सीडलिंग पे पड़ रही है उसके अपोजिट साइड पे मिका की प्लेट को रखा, एंड दे ऑब्जर्व दैट जो ऑक्सिन था वो यहाँ पे ट्रांसफ़र नहीं हुआ और सीडलिंग ने कर्वेचर शो नहीं किया. बट व्हेन दिस मिका प्लेट वाज इंसर्टेड इन द डायरेक्शन ऑफ लाइट, सीडलिंग में उस डायरेक्शन में जहाँ पे लाइट पड़ रही है, सो जो ऑक्सिन होता है वो शिफ्ट हो जाता है लाइट वाली साइड से डार्क साइड में, और वहाँ पे सेल इलोंगेशन करता है, जिसकी वजह से जो सेलिंग है कर्वेचर शो करती है इन द डायरेक्शन ऑफ लाइट. देन इन नेक्स्ट स्टेप व्हाट दे हैव डन, they inserted gelatin block in the excised portion between coleoptile and the rest of the seedling. और उन्होंने ऑब्जर्व किया कि जो टिप थी coleoptile की था, वहाँ से जो ऑक्सिन है दैट इज़ डिफ्यूज्ड इनटू द सब एपिकल रीजन, और ये शो करता है कर्वेचर व्हेन इट रिसीव द लाइट स्टिमुलस. सो दिस ऑक्सिन कैन डिफ्यूज थ्रू दिस gelatin block. Next experiment was carried out by Avena. So he used this agar gel block. Firstly उन्होंने जो कोलियोप्टाइल है उसको अगार ब्लॉक के ऊपर रखा, जो ऑक्सिन है इससे वो इसमें ट्रांसफ़र हो गया, एंड देन दे पुट दिस अगार ब्लॉक ऑन द सीडलिंग and it shows the same response as that of the coleoptile. So they finally concluded that this coleoptile or tip of the plant is responsible for the formation of that chemical substance which causes the unidirectional growth or curvature in the direction of light. So FW Wendt was a scientist जिन्होंने एक एक्सपेरिमेंट किया जिसका नाम है Avena coleoptile curvature test. This is also known as the bioassay for Auxin. So FW Wendt isolated tip of oat. Oat is a member of family Poaceae, commonly known as Avena, botanical name is Avena sativa, and put that coleoptile on agar plate. So test was named as Avena coleoptile curvature test. It is also known as bioassay of Auxin. Bioassay means to know the power of any chemical substance produced by a living organism. So this experiment suggested that auxin is synthesized in the coleoptile tip. इस एक्सपेरिमेंट से ये पता चला जो ऑक्सिन है वो coleoptile की टिप में बनता है and is translocated downwards. नीचे की डायरेक्शन में ये ट्रांसपोर्ट होता है, that is basipetal movement of this chemical substance. So in this tip we can see here, so this pinkish color shows the presence of auxin in the tip. When this tip is excised, जब ये टिप को एक्साइज किया जाता है and और इसको अगार के ब्लॉक के ऊपर रखा जाता है, so this auxin gets diffused into this agar block. और जब इस अगार ब्लॉक को decapitated stump पे रखा जाता है, so auxin containing agar block in one side of the stump, this auxin gets diffused into the lower portion of the seedling, जिससे जो auxin है नीचे की तरफ मूव करता है और यहाँ पे सेल की ग्रोथ होती है, and it shows growth in one direction and show curvature in the direction of light. So he discovered that जो 35% ऑक्सिन है ये प्रेजेंट होता है इल्यूमिनेटेड साइड पे, मीन जहाँ पे लाइट स्ट्राइक करती है सेडलिंग पे, और जो 65% है ऑक्सिन दैट इज़ प्रेजेंट ऑन द डार्क और शेडेड साइड. दैट इज़ ये जो इल्यूमिनेटेड साइड है वहाँ से ऑक्सिन मूव करता है टू द शेडेड साइड, और ड्यू टू दिस वहाँ पे ऑक्सिन की कंसनट्रेशन ज़्यादा होती है, जिसकी वजह से जो सेल्स हैं वो ज़्यादा इलोंगेट होते हैं, एंड दैट्स द रीज़न टू बेंड इन द डायरेक्शन ऑफ light source. So it shows positive phototropism where faster cell elongation is seen on the shaded side and less on the illuminated side. So this was all about this FW Wendt's Avena coleoptile curvature test. The picture which again shows these three experiments, experiment by Darwin and his son, then Boyce and Jensen, and finally the Paul experiment and the experiment which was carried out by Wendt in case of Avena sativa. Next are types of auxins. Basically दो तरह के auxins हैं, one है natural auxin और other are synthetic auxin. So auxin जो plants में naturally पाए जाते हैं उनको बोला जाता है natural auxin. For example, indole 3-acetic acid, indole propanoic acid. In some book this compound is not mentioned. Then indole butyric acid and phenyl acetic acid. So these are naturally occurring auxins in plants. Then some auxins which are synthesized artificially but they shows similar functions as that of the natural auxins. इसमें हमारे पास example है 2,4-D, that is 2,4-dichlorophenoxyacetic acid, then 2,4,5-trichlorophenoxyacetic acid, and naphthalene acetic acid. So these are some synthetic auxins and they play a very important role in agriculture. So these are the chemical structures of natural auxins: indole 3-acetic acid, and 4-chloro indole 3-acetic acid, indole 3-butyric acid, and these two are the structures of this synthetic auxin like 2,4-D, and in case of 2,4,5-T, so there will be one more chlorine here. Then there is other synthetic auxin जिसका नाम है 2-methoxy-3,6-dichlorobenzoic acid, commonly known as dicamba. So these are some synthetic auxins chemical structures. Next are antioxins. So as the name indicate, these are the chemical substances which work against auxin. So group of chemicals that prevent the auxin action in plants. जो auxin के action को prevent करते हैं उनको antioxins कहते हैं. Discovered by Skoog in 1942. So some chemicals like tranexamic acids and ascorbic acids act as antioxins. So there are some inhibitors of auxin transport, chemical substances जो auxin के transport को inhibit करते हैं. For example, TIBA, TIBA, which stands for 2,3,5-triiodobenzoic acid. So this is the chemical structure of this TIBA. Next, auxin transport inhibitors with their chemical structures. So these are some auxin transport inhibitors which are not found in plants, जो कि plants में नहीं होते हैं. For example, NPA, that is naphthylphthalamic acid. Then TIBA, 2,3,5-triiodobenzoic acid, and NOA, that is 1-naphthoxyacetic acid. So these are the chemical substances which inhibit the auxin transport. Then there are some inhibitors जो कि naturally पाए जाते हैं plants में. For example, quercetin, that is a flavanol, and gencitin. So these are the natural occurring inhibitors of auxin transport. Next are the physiological roles of auxin. First, the first role is in cell elongation. Auxin plays a very important role in cell elongation. So it stimulates the elongation of cell in shoots. So auxin causes cell elongation probably by it may activate proton pump and export the protons out of the cell, जिसकी वजह से pH low हो जाएगा. By this, it increases the osmotic concentration of solute in the cell, जिससे cell में इसकी जो concentration है वो increase हो जाएगी. And this results in activation of wall loosening enzyme also, जिससे cell wall loose हो जाएगी. And due to increase in osmotic solutes, so there will be endosmosis from the surrounding cell, जिससे जो cell have that become turgid and exert pressure on the cell wall. और wall lose होने की वजह से जो cell wall है उसमें जो wall pressure है वो reduce हो जाता है. Then it also increases the permeability of the cell to water. So that's why जो surrounding cell है, जो water है, उससे cell के अंदर enter करेगा through endosmosis. So this is the picture which shows cell elongation. Suppose this is the auxin, this is the cell. It causes export of proton out of the cell, जिससे यहाँ पे जो osmolytes है उसकी concentration बढ़ जाएगी. और जब osmolyte की concentration यहाँ पे बढ़ जाएगी, solutes की concentration बढ़ जाएगी, so water जो surrounding cells से हैं इसके अंदर enter करेगा through endosmosis. और it also causes the loosening of the cell wall. जैसे पानी इसके अंदर enter करेगा, so it will apply pressure from inside to outside, जिससे expand होगा, and this results in elongation of cell. So by an increase in the cell wall synthesis and including synthesis of some specific DNA dependent new mRNA and specific enzymatic proteins. So ये जो सारे constituent particles हैं वो बनते हैं, जिससे कि cell wall जहाँ पे lose होती है, जहाँ पे gaps बन जाते हैं, वहाँ पे नई cell wall के components को synthesize किया जाता है. And this brings increase in cell plasticity. So plasticity means growth जो होगी इसमें that is not reversible, so it will be permanent growth. Next, very important feature or physiological role of auxin in apical dominance. So it is the suppression of lateral bud in the presence of apical bud. For example, जो plant है जिसमें ये apical portion present रहता है, उसमें जो lateral buds हैं they are not able to show the growth. So जो ये suppression है lateral buds का in the presence of this tip, this is known as apical dominance. जब इस tip को remove कर दिया जाता है, So the lateral buds and they will show growth. This is known as lateral dominance. So many hormones play important role in this phenomenon. So this is the suppression of lateral growth by apex because of the presence of apical bud. So apical bud is the source of auxin. Skoog and Thiman in 1934 first pointed out जो apical dominance है that might be due to the presence of auxin or under the control of auxin. बट अगर हम टर्मिनल शूट ऑफ द प्लांट को रिमूव कर देते हैं, अगर टर्मिनल पोर्शन को रिमूव कर देते हैं, जो लेटरल बर्ड्स हैं उनकी ग्रोथ स्टार्ट हो जाती है. सो इन दिस पिक्चर वी कैन ऑब्जर्व हियर इफ दिस सीडलिंग इज डीकैपिटेटेड मीन्स इसके टिप को रिमूव कर देते हैं तो जो लेटरल बर्ड्स हैं वो ग्रोथ शो करते हैं. सो गार्डनर जो होते हैं दे यूज दिस प्रिंसिपल बाय प्रूनिंग द टर्मिनल शूट्स, जिससे जो लेटरल बर्ड्स हैं वो ग्रोथ शो करते हैं. So auxins inhibitory effect on lateral bud is not direct. ये direct effect नहीं है, but it is indirect, possibly through the involvement of other hormones like cytokinins and abscisic acid. So lateral जो birds हैं उसमें दो और हार्मोन important role play करते हैं, जो cytokinin है और ABA, which plays an important role in this dormancy of lateral bud that we will know in upcoming slides. So the application of cytokinin to the lateral bud stimulate bud growth in many plants. अगर हम lateral side पे cytokinin को apply करते हैं, so उससे ये जो birds हैं lateral birds they will show growth, and the ratio of auxin to cytokinin is the key factor which is involved in the apical dominance. So ratio auxin का और cytokinin का decide करता है कि जो apical dominance है वो होगी या नहीं होगी. If the concentration of auxin is more and the concentration of cytokinin is low, so that will promote apical dominance, means lateral buds dormant रहेंगे. But when the concentration of cytokinin is more and concentration of auxin is less, तो उस time पे lateral buds growth show करेंगे. So this picture again shows this phenomenon. In the presence of this tip, it will grow upward direction. This को बोलेंगे हम apical dominance. But अगर हम इसको remove कर देंगे tip को, then it will show the lateral growth of lateral buds. So it will form a bunch of branches. So higher auxin to cytokinin ratio suppresses the growth of lateral buds. Means it causes apical dominance. जब auxin concentration ज़्यादा होती है. So ABA is well-known growth inhibitor, has been observed in the dormant lateral bud. ये देखा गया है कि जो lateral buds हैं in the presence of auxin यहाँ पे ABA present रहता है. So auxin is responsible for maintaining the concentration of ABA in the lateral buds. So ABA करता है dormancy cause in lateral buds में. But when we remove this tip, so after removal auxin का source degrade हो जाता है, and that also leads to degradation of this ABA concentration, जिससे finally ये जो buds हैं वो grow करते हैं. So in intact shoot, auxin helps to maintain the higher concentration of ABA in the lateral buds. So that results in the inhibition of lateral buds growth. Next physiological role of auxin is root initiation in stem cuttings. So higher concentration of auxin inhibit the elongation of root and initiates the growth of lateral roots or adventitious roots. जो मेन रूट है प्राइमरी रूट उसकी ग्रोथ को इनहिबिट करता है हायर कंसंट्रेशन ऑक्सिन का, बट जो लेटरल रूट्स हैं विच आर फॉर्म फ्रॉम पेरिसाइकल उनको ये इनिशिएट करता है. सो व्हाट ऑक्सिन डू, ऑक्सिन स्टिमुलेट करता है पेरिसाइकल के सेल्स को टू डिवाइड एंड रिजल्ट्स इन द फॉर्मेशन ऑफ लेटरल रूट. So application of indole acetic acid in lanolin paste to the stem cuttings or cut end of the young stem can result in the formation of extensive roots or adventitious roots, and this method is of great practical importance. ये जो method है बहुत ही important है for the practical use and to initiate rootings in the stem cuttings of widely utilized economically important plants which are propagated by stem cuttings. ऐसे economically important plants जिनको हम stem cuttings के थ्रू propagate करते हैं, So we can use this method to initiate root system in those stem cuttings. So this is the picture. So this picture, when we put the stem cutting in the Auxin solution, so it can result in the formation of these lateral roots or adventitious roots. So this is the important use of Auxin in root initiation. Next is leaf abscission. So Auxin is a growth promoting hormone. तो ये leaf abscission को stop करता है या रोकता है. Auxin control the formation of abscission zone. Auxin mainly abscission zone बनने नहीं देता है. In older leaves, जो older leaves होती हैं उनमें Auxin की concentration कम होती है, means Auxin की synthesis कम होती है as compared to young leaves. So that's why जो old leaves होती हैं उनमें abscission के chances ज़्यादा होते हैं. So higher content of Auxin in the leaf lamina prevent the formation of this zone, which is known as abscission zone, responsible for leaf abscission. And the low concentration of Auxin accelerate the formation of abscission zone. अगर concentration Auxin की कम है, जो कि mostly mature leaves में होती है, so this will accelerate the process of formation of abscission zone and finally results in the abscission of leaf. And this phenomenon was first observed by Liebesch in 1933. So this is the picture. This is the leaf lamina. So जो mature leaf है उसमें Auxin की concentration कम होती है. अगर Auxin की concentration कम होती है, so यहाँ पे जो petiole है इसके base पे जो ABA है that will increase and it results in the formation of this zone of dead cells जो कि thin walled cells होते हैं. And this zone is known as abscission zone, and this is responsible for the abscission of this leaf. So Auxin have important role to prevent the formation of this abscission zone. So Auxin होता है, अगर young leaf है तो Auxin concentration ज़्यादा होती है, so it moves towards the petiole and prevent the formation of this abscission zone. But when Auxin synthesis is poor, जो mature leaves होती है Auxin की synthesis कम होती है, तो वहाँ पे जो ethylene है और ABA है वो ज़्यादा बनता है और वो responsible होता है formation of this abscission zone. Finally ये leaf यहाँ से senescence के बाद इसकी abscission हो जाती है, means ये जो leaf है it is shut down from the parent plant. Next is flowering. Auxin also plays important role in inducing flowering. So Auxin treatment can induce flowering in plants like pineapple and also promote the formation of female flowers, especially the pistillate flowers in case of dioecious plants. And the low concentration of this NAA, that is naphthalene acetic acid, or indole acetic acid, are also effective in inducing flowering in barley. So barley में ये जो दो chemical substance हैं, hormones हैं, they are effective in inducing flowering. While 2,4-D, that is 2,4-dichlorophenoxyacetic acid, that controls the flowering in sweet potato. So this is the pineapple. So this hormone, that is the Auxin, it induces flowering in case of pineapple. Auxin also plays very important role in fruit development. So helps in development of fruits. Stimulate development of fruit by controlling size. So in case of strawberries, जो strawberries होती हैं उनमें कुछ एक seed like structures होते हैं जिनको कहा जाता है achenes, and they are thought to be the source of Auxin. इनको Auxin का source माना जाता है. अगर strawberry से इन achenes को remove कर दिया जाए तो जो strawberry का fruit है उसकी development complete नहीं होती है, that becomes greenish in color और उसका size भी reduce हो जाता है. But अगर हम उस स्ट्रॉबेरी में जिससे एकीन्स रिमूव किए हैं उसको ऑक्सिन की ट्रीटमेंट देते हैं एक्सटर्नल, सो दैट विल डेवलप नॉर्मली. सो दिस इज द पिक्चर ऑफ दिस फ्रूट और स्ट्रॉबेरी. सो दीज आर द स्ट्रक्चर्स विच आर नोन एज एकीन्स और ये है सोर्स ऑफ ऑक्सिन. अगर इन एकीन्स को रिमूव कर दिया जाए तो जो साइज होता है फ्रूट का दैट रिमेन्स स्मॉलर इन साइज और ये प्रॉपरली ग्रो नहीं करता है. बट व्हेन दिस फ्रूट इज सप्लाइड विद ऑक्सिन, अगर इसको ऑक्सिन की ट्रीटमेंट देते हैं एक्सटर्नली, सो इट विल ग्रो नॉर्मली, इट विल इंक्रीज इन साइज एंड शो नॉर्मल डेवलपमेंट. सच फ्रूट्स ऑन ट्रीटमेंट विद ऑक्सिन शो नॉर्मल डेवलपमेंट. एक ऑक्सिन का और फीचर है दैट इज़ वेरी इंपॉर्टेंट. ऑक्सिन हेल्प्स इन फॉर्मेशन ऑफ पार्थेनोकार्पिक फ्रूट्स. So these are the fruit which are seedless. For example, in the members of family Solanaceae and Cucurbitaceae. So we can see this is the member of Cucurbitaceae, watermelon. So this watermelon is seedless. So we can see this is a parthenocarpic fruit जो कि ऑक्सिन की ट्रीटमेंट से बनते हैं. ऑक्सिन also control the premature fruit dropping. जो premature fruit dropping होती है, that is also controlled by auxin in case of fruits like apple, and it also improves the quality of fruit, helps in increasing the quality of fruit. Next is callus formation. Callus is an undifferentiated mass of cells, so may activate cell division. Continuous growth of callus takes place only after the addition of auxins. So this is the picture of callus. So, callus is a group of cells which are undifferentiated. That means they are continuously dividing cells. Then it also helps in vascular differentiation. When plants suffer from mechanical injury, अगर किसी plant में कोई injury होती है तो जो vascular region का repair होता है that is also done with the help of this auxin. So application of auxin promotes the formation of xylary element or trachy element. जहाँ पर damaged वेस्कुलर सिस्टम होता है उसकी डिफ्रेंसिएशन, उसकी फॉर्मेशन के लिए ऑक्सिन हेल्प करता है. एंड द प्रोसेस ऑफ वेस्कुलर डिफ्रेंसिएशन इज अंडर द कंट्रोल ऑफ बोथ ऑक्सिन एंड साइटोकाइन. सो दिस प्रोसेस इज कंट्रोल्ड बाय बोथ द हार्मोन्स ऑक्सिन एंड. दिस पिक्चर शोज द रोल ऑफ ऑक्सिन इन वेस्कुलर डिफ्रेंसिएशन. दिस इज अ cucumber's plant in which the apical bud is intact. और this जो apical bud है, this is the source of auxin. When this apical bud is removed, the supply of auxin is interrupted. Then in the seedling एक wound create किया जाता है और wound के ऊपर वाला जो portion है उसमें जो leaves present होते हैं they are also removed. And after the removal of leaf there is complete stoppage of auxin supply. जो ऑक्सिन की सप्लाई है नीचे की तरफ को वो रुक जाती है. देन आफ्टर दैट इन द इंजर्ड साइट इफ़ वी अप्लाई दिस इंडोल एसिटिक एसिड अलॉन्ग विद लैनोलिन पेस्ट, सो ये जो वाउंडेड पोर्शन पे जब इसको रखते हैं सो इस वाउंडेड पोर्शन में ये जो ऑक्सिन है वेस्कुलर डिफ्रेंशिएशन मीन्स जाइलम जो एलिमेंट्स हैं ट्रैकरी एलिमेंट्स उनकी फॉर्मेशन स्टार्ट करता है. सो दिस कैन बी ऑब्जर्व्ड अंडर माइक्रोस्कोप in this way. So auxin helps in vascular differentiation, means it helps in synthesis of this trachy elements in the wounded plant part. So auxin plays very important role in vascular differentiation. Next, role of some synthetic auxins in various fields. Firstly, synthetic auxins are used as weed killers. Include 2,4-D, that is 2,4-dichlorophenoxyacetic acid and 2,4,5-trichlorophenoxyacetic acid. They are the weed killers and they are used as a herbicide against the broad-leaved herbs. So used as selective herbicides against broad-leaved plants. And they also helps in tolerating the frost damage. जो चिलिंग इंजरी है उससे टॉलरेट करने के लिए भी ये जो 2,4,5-ट्राइक्लोरोफेनॉक्सिएसिटिक एसिड हेल्प करता है इन एप्रिकॉट फ्रूट्स. देन weapons of war. So these synthetic auxins were also used in war. The war was between Vietnam and US. So US military used one chemical substance जिसका नाम है Agent Orange. And this Agent Orange chemical composition is it consists of 1:1 of 2,4-D and 2,4,5-T. So this is a defoliating agent. It destroys the crops and the forest vegetation. So this is all about the auxin. The next category of hormones is gibberellins or GAs, gibberellic acid or gibberellic acids. So these are also growth promoting hormones which helps in cell elongation and internal growth, and they are also associated with the germination of seeds. So Gibberellins, this is the chemical structure of gibberellic acid, that is GA3. Gibberellic acids की बहुत सारी varieties present होती हैं. So they are the chemical substances which are approximately 225 to 130 in their numbers, and this is a 20 carbon compound and it's commonly known as diterpene. So mostly it regulates the plant height की height को regulate करता है, and it also controls seed germination. So these are two important events related to this hormone. It is a large group of related compounds, and it's the only group of plant hormones which are described on the basis of their chemical structures rather than their biological activity. So this is the only chemical substance जिसको हम differentiate और describe करते हैं on the basis of its chemical structure rather than its biological activities. So बहुत सारी variety of chemicals present होते हैं gibberellic acid. They are of different types. Then discovery. So firstly, it was discovered by a person named as E. Kurosawa in Japan. He observed a plant which was infected with the fungal pathogen. एक fungal pathogen है जिसका नाम है Gibberella fusicari, and this fungus pathogen is responsible for causing one disease in the rice plant जिसको बोलते हैं Bakane disease or foolish seedling disease of rice. So this is the rice field. In this rice field we can observe these some rice plants which are pale in color, और इनकी जो height है that is much longer as compared to the normal plants. So this diseased form is known as Bakane or foolish seedling disease in case of rice. So it was first observed by E. Kurosawa. Then this substance causes the plant to grow speedily tall and pale in color, that we can see in this picture. The next, there was two scientists, T. Yabuta and Y. Someki. They were the first person to isolate this substance from this fungus, और इसका नाम रखा था उन्होंने Gibberellin. Then 1955, other persons, Stodola and Bryan, isolated this gibberellic acid from this fungus. Gibberellic acid was first isolated by these two persons from this fungus. Then in 1956, West and Pine, they isolated GA from the plants, other plants. So it is present in all types of plants. Next, in 1958, Macmillan in Bristol, England, identified GA1 from beans. So they isolated this chemical substance from beans. And currently, there are more than 125 to 130 gibberellic acids which have been isolated, जो कि आज तक isolate किए गए हैं. And out of these, 24 types के gibberellins सिर्फ और सिर्फ एक ही fungus Gibberella fusicari isolate किए, and rest are one and one are from other higher plants. Then GA3, which is a 20 carbon compound, is also known as gibberellic acid. GA3 को gibberellic acid भी कहा जाता है. And GA1 and GA20, these are both carbon 19 compounds and are most active forms and most important forms. So ये दोनों forms बहुत active forms होती हैं GA की. Then gibberellic acid is easily available from fungus. Fungus से easily इसको isolate किया जा सकता है. And chemically it is a diterpenoid, mainly control the plant height and seed germination that we have discussed in the starting slide. So these are some structures of GAs. So this is the active form of this GA, and this is the inactive form of this GA. This is the 19 carbon containing gibberellic acid. So this is GA1 and this is GA8. GAs plays important role in elongation of shoots. So they help in cell elongation and improve internodal growth. So stimulate cell elongation similar to Auxin, and they also promote internodal growth in plants. Some plants which are genetically dwarf means they lack the gene for the synthesis of GA, and some plants which are treated with some inhibitors of GA, they show stunted growth. When they are treated with exogenous application of gibberellin, so they show improvement in their growth. So plants with short internodes may not have the gene for gibberellin synthesis. And inhibition of stem growth by some inhibitors like alar, phosphorin, and AUXINS, which are the inhibitor of GA. So GA exogenous application overcome the effect of these inhibitors as well as the deficiency of gene which is responsible for gibberellin synthesis. So ये दोनों केसेस में ये जो एक्सटर्नल एक्सोजिनस एप्लीकेशन है जीए की वो ओवरकम करती है इनहिबिटरी केमिकल्स के इफेक्ट को भी, और अगर किसी प्लांट में जीन नहीं है जिबरेलिन सिंथेसिस की, so by applying external GA the plant will show the normal growth. So in this picture we can see here, this is the pea seedling which is without GA, and this is the pea seedling with the application of exogenous GA3 gibberellic acid. So they show enhanced stem growth or elongation of internodal growth as compared to this control plants. ये बहुत ज़्यादा growth show करते हैं when the plants are treated with GA gibberellic acid, which proves that gibberellin promotes stem elongation in many plant species. Next, elongation in dwarf and rosette plants. So GA plays important role in the formation of stem in rosette plants. So many herbaceous plants show rosette habit, that is जो leaves होती हैं they are arranged in circular form and they are at the same height. So this arrangement is just similar to that of rose flower. जैसे कि petal की arrangement rose flower में होती है, वैसे ही इन plants में leaves की arrangement होती है, और जो stem होता है इन plants में that is highly reduced. So this is due to deficiency of gibberellins. So the treatment of these plants with external gibberellic acid or gibberellic acid induces bolting. So this is the very important phenomenon shown by this GA. Bolting means rapid shoot elongation. So in rosette plants, when there is rapid shoot elongation, that process is known as bolting. So genetic dwarfism in many mutants like Pisum sativum, Vicia faba, which is commonly known as broadbean, have been overcome by the application of this GA3 gibberellic acid. So they play an important role in elongation of stem or shoot in genetically dwarf plants and in rosette plants, and this phenomena of elongation of shoot in rosette plant is known as bolting. So this is unique feature of GA. So in this picture we can see here, this is the plant which is a rosette plant having highly reduced stem, और जो leaves होते हैं they are arranged at a single point, and this arrangement is similar to the petals in rose flower. But when this plant is treated with external GA, it will show elongation of the stem, and cause flowering in these plants also. So this rapid growth in rosette plants in the shoot is known as bolting. This phenomenon is known as bolting. Next is flowering. So treatment of this GA gibberellic acid in long day plants under short day condition. अगर हम long day plants को short day conditions में GA की application देते हैं, so they induce flowering. So we can overcome the long day condition under short days, long day requirement under short day condition by treating the plants with gibberellic acid. So it also replaces the cold treatment, which is the vernalization, and long day photoperiodic treatment in many plants. For example, this is a plant which is known as Hyoscyamus niger, जो कि एक long day plant है, but इसमें flowering को short days condition में induce कर दिया जाता है by the application of GA, gibberellic acid. So this is without GA, this plant shows rosette habit, but when it is treated with GA, gibberellic acid, it will show this bolting phenomenon, and also there is formation of flowers. So we can overcome the requirement of long days under short days condition in long day plants by applying external GA. So it also induces flowering in plants which are long day plants under short days condition, and also replaces the requirement of cold treatment, that is vernalization. जो vernalization है उसको भी ये replace करता है. Next is parthenocarpy. Partheno means virgin, carpy means fruit, producing fruits which are seedless. GA is known to induce parthenocarpy just like to that of Auxin in many plants like Cucumis sativus, Solanum melangena, and Zaphanthus. So this is the tomato fruit which is without seeds. So this type of this type of fruit which is without seed is known as parthenocarpic fruit. This is brinjal, that is Solanum melangena, which is again without seeds. So this GA is known to produce parthenocarpic fruits, and this is the plant of Zephyrinthus, which is a ornamental plant. But this GA is more effective than Auxin, and seedless fleshy tomatoes with large size grapes are produced by using this gibberellin. So we can produce very good quality tomatoes and grapes by using this gibberellin. Next is seed dormancy. This GA plays very important role in breaking the seed dormancy. So it is known to break seed dormancy. What it does, it induces this aileron layer in the seed. In case of cereals, जो ये aileron layer होती है, it is made up of proteins. It activates this aileron layer and produces certain enzymes which are responsible for the breakdown of carbohydrate like amylases and some proteases which helps in breakdown of proteins. So जो amylases हैं ये breakdown करते हैं starch को into simple sugars, और वो जो simple sugars होती हैं they are used by the embryo for its growth. It induces seed germination in light sensitive seeds of tobacco and lettuce in complete darkness. So GA is known to induce seed germination under dark condition in light sensitive seeds, or we can say photoblastic seeds. So these are the seeds of lettuce, and this is the plant of lettuce, which is commonly known as salad. Seeds of temperate species like apple, peach require low temperature and can be replaced by GA for seed germination. So the germination of seeds in case of some temperate plants like apple and peach, they require cold treatment for the germination of seed. So वो जो cold treatment है इन plants में वो replace की जा सकती है by the external treatment with GA, gibberellic acid. Next is bud dormancy. This helps in overcoming the bud dormancy in the buds of temperate region due to severe cold conditions. So these are the buds which are dormant. So when they are treated with this GA, then these becomes active and form the shoot or leaves. So potatoes also have dormant phase after harvest. By the application of GA sprouts eyes vigorously. So बहुत ही फास्ट ग्रोथ करते हैं अगर हम एप्लीकेशन कर देते हैं GA की externally. So dormancy in apical buds of peach is also reversed by GA. So these are some uses of GA to overcome the bud dormancy. Next is senescence. It is the gradual programmed cell death or aging process. So GA3 application delays the senescence of fruits and also prevent the senescence of leaves in plants like Rheomex and Tropiolum. So this is the picture which shows the gradual degradation in the chlorophyll. जो chlorophyll है, chloroplast है, वो degrade हो जाता है, and this causes the yellowing of leaves, finally the death of leaf, which is known as senescence or aging. Next is seed germination in cereals that we have already discussed. It is essential for this GA is essential for the germination of seeds in case of cereals. Its exogenous application stimulates the aileron layer and causes the release of certain enzymes like amylases and proteases. So जो GA है ये aileron, this is the aileron layer, यहाँ पे ये activate करता है certain enzymes को like alpha amylases and proteases जो break down करते हैं starch को और proteins को, और जो solubilized form है for example like glucose and maltose. So these soluble forms are used by embryo for its growth, और इस तरह से seed germinate करेगा. So enzyme participate in the breakdown of stored starch to the simple sugars and transport to the developing embryo, which results in the growth of that embryo, जिससे कि embryo की growth हो जाएगी और जो seed है वो germinate करेगा. Next is seed germination. So light sensitive seeds, which are commonly known as photoblastic seeds, for example lettuce and tobacco, they show poor germination under dark condition. अगर उनको हम dark condition में germinate करते हैं, so they show poor germination. But under dark condition, if we apply some external gibberellin, so they will show the proper germination. So they germinate on exogenous treatment with GA in dark. So dark condition में भी अगर हम exogenously treatment देते हैं gibberellic acid की, so वो show करेंगे germination. So this is the lettuce seed which is showing the germination. Next is sex expression. So GA plays important role in sex expression, just like that of auxin. It induces maleness in the flowers and promotes the formation of male flowers on genetically female plant of cannabis and also in cucurbits. So in this picture we can see here, this is the male flower and this is the female flower of cucurbita. So in the plant which is genetically equipped or genetically made for producing female flowers, अगर हम उसमें external GA की application देते हैं, so वो जो plant है that will produce only male flower. So this was all about today's discussion about plant hormones, auxins and gibberellins. Hope you will get some idea from this presentation. If you have any questions, queries and any suggestions, you can give it in comment section. Thank you. Have a nice day.